Great, thank you very much for having me. I'm now working at UCL at the Wellcome Center for Human Neuroimaging, but I spent a lot of this work was done a lot at Montreal and also in Jülich. And it's work presenting the BigBrain atlas of cortical layers, which you've seen a few images of today. And the work was aimed to link cortical microstructure to in vivo measures of cortical structure. So the BigBrain is uniquely placed to act as a bridge between MRI and histology. For MRI, we can measure with large numbers of individuals, and in vivo, we can measure function as well as structure. But generally speaking, it is of low resolution with an ambiguous signal. What I mean here is if you have a difference in cortical thickness, this could be driven by differences in myelination of the cortex or differences in the thickness of different layers, and we can't really say which at the moment with MRI. At the other end of the spectrum, we have histology, where we have very high resolution, and we can be very specific about what we're staining or labeling. Generally, this has been 2D with small numbers of individuals and very limited numbers of aerial samples, as well as requiring a large amount of manual annotation as well. And so the BigBrain as a 3D digital histological atlas is uniquely placed to provide a link between these two modalities and scales. So in the talk, I'm going to cover how we generate laminar intensity profiles from the BigBrain, how we use these profiles to automatically segment cortical layers in the BigBrain, and then ultimately this atlas of cortical layers, which we now have for the isocortex. Don't really need to introduce the BigBrain as a data set. Suffice it to say that if you look at panel H here on the bottom right, it is 20 micron resolution data, which is sufficient to pick out laminar features within the cortex, which is crucial to the work that we're trying to do here. So the first step of this work to extract the profiles was to create cortical surfaces. Much as you would within MRI, it involves extracting a white matter surface and then a peel surface. And the key point here is that every point on the peel surface has a corresponding point on the adjacent white matter surface. This was work done by Claude Lepage and Lindsay Lewis. What this enables you to do is to sample the intensity of the cortex of the BigBrain at 100 points between the peel and white matter surfaces. And this enables you to create an intensity profile at each vertex on the surface. So here's one example. The black arrow is the streamline of that intensity profile. And in the top right, you can see, can I minimize this? On the top right, you can see the intensity. And on the bottom right, you can see the first derivative of that same intensity profile. And you can see that there are peaks and troughs that correspond to the laminar features you can see in the 2D image to the left. In order to segment layers, you need to find features within these profiles that are consistent across the profiles. And the first really obvious feature that we noticed was the boundary between layers 1 and layer 2. And this is the pale area going to the dark within the cortex. And on the first derivative, you can see this large peak in the gradient of the intensity profile. So if you identify this peak in all the profiles, you'll end up with a surface that follows this feature that you can see in the image. The second feature that was relatively consistent across profiles was this peak in the center of the cortex, which is this dark band on the histology. And this roughly corresponds to layer 4. Again, if you can identify this in each of the profiles, you end up with a surface layer that will follow these features. Once you have an algorithm that can find these consistent features, you can apply it to all the profiles across the cortex. And this creates 3D surfaces, comprehensive surfaces for this layer 1, 2 boundary and layer 4. So this was the first piece of work we did, identifying laminar structure and laminar features within the cortex. And one of the things we noticed, which was really nice to see, was that this layer 4 surface, if you look in a sulcus up here on the top right, is very deep and close to the white matter surface. Whereas in the adjacent gyrus, it's much more superficial, much closer to the peel surface. But now that we have this 3D volume, we can quantify this effect. So we can measure the curvature of the surface, which describes if the curvature is positive, we're in a gyrus, and if the curvature is negative, we're in a sulcus. You can see that across the cortex, within the sulci, layer 4 depth is much greater, so it's 70% depth in the bottom of sulci, whereas in the adjacent gyri, it's much more superficial, it's blue or green, maybe 40–50% depth. What's nice is there's a model called the Equivolumetric model, which states that within a gyrus and the adjacent sulcus, a layer will take up the same amount of volume. So if you're on the outside of a bend, you'll be stretched and much thinner. And if you're in the inside of the bend, you'll be compressed and your layer will be much thicker. And you can calculate this equivolumetric depth from the peel and white matter surfaces alone. You don't need any of the surfaces in between to calculate this. And if you calculate this, you find that this layer 4 surface is very consistently at about 60% cortical depth, i.e. you're accounting for the curvature relationship very well with the equivolumetric model. So now that it's been thoroughly validated in 3D on histology, we're able to implement this. You're able to generate these equivolumetric surfaces from just the peel and the white matter surface. So we made a package that can do this. And what's nice is you can generate these surfaces from MRI. So from the peel and white matter surfaces of a free surface reconstruction or a civet reconstruction, you can generate these equivolumetric surfaces. And this is freely available on GitHub to use. So here, the yellow surface is a mid-thickness surface, not equivolumetrically derived, and the red lines are the equivolumetric surfaces. And the yellow line crosses over many layers between the folds, whereas the red lines follow consistent laminar features between the folds. So now that we have profiles that indicate laminar information, we want to create a full 6-layered atlas of the cortex. And this requires slightly more flexible learning algorithms to identify these layers. The main problem is that cytoarchitecture varies from area to area, and also the features that define cortical layers aren't always clear peaks and troughs. Sometimes they're more subtle features. Deep learning is really well placed to address this problem because rather than having to hand code the features you're interested in, the convolutional neural networks will learn filters that are useful for identifying features that you want in the image. The problem is often this requires really large training data sets. And so rather than using the 2D convolutional neural networks that are very typical, so for instance, here below, identifying cats and dogs with a 2D convolutional network, we keep with this 1D profile-based approach. And this enables us to generate a very large training data set. So we manually segmented 60 training regions at a variety of positions within the big brain, left and right hemisphere, different cytoarchitectures, front and back of the brain. And then from each of these, here in panel A, you can see 2 of these manually segmented regions. And from each of these, we can derive many, many training profiles. You can just extract many parallel profiles, and this means they can be labeled according to the manual annotation. You can extract the intensity values of the big brain, and you can create over 500,000 labeled training profiles from 60 annotated regions. And here's 2 examples of these profiles on the right. You've got a raw profile here, a smooth profile here, and then the ground truth labeling from the manual segmentations on the bottom. And this is a second profile. And you can then show the network these 500,000 profiles, teach it to learn features that correspond to these layer boundaries, and then run it on the full big brain data set. And what you end up with is a segmentation of the 6 cortical layers across the cortex, which is the images that have been shown earlier today and are shown here. One feature to draw your eye to is that in the top left is the coronal section, which is the original plane of acquisition. And where the crosshairs are is this weird island of layer 3, this orange blob, which seems to make no sense. And you certainly wouldn't have been able to identify it as that with the in-plane 2D histology alone. However, because the big brain is this 3D histological data set, and we're using a 1D profile-based approach, in the 2 other planes, you end up with very plausible segmentations of the 6 layers. And it's just that the cutting plane has really obliquely sectioned the cortex in the coronal plane. This data is now freely available from the Big Brain website and is also part of the Human Brain Project Atlas. And you can browse this online. I think Timo may have shown this earlier. I'm just going to draw to a couple of features that I really like about these results. So here are the cortical layers. Here they're colored lines instead of the dense pixel segmentation. And the gray is where are two areas of manual segmentation. So one thing that you can see is that the layers follow what is drawn manually pretty well. And also that beyond the extent of the manual segmentations, it still seems to be identifying the same features on previously unseen cortex. The second thing is that if we zoom in here, this is the V1, V2 boundary, so primary visual cortex and secondary visual cortex. And at this point, layer 4, which is between the green and blue lines, goes from being relatively thin in V2 above to being very thick and multi-sublayered in V1 here. And the network is able to identify from individual profiles whether it's looking at a V2 or a V1 profile and gives the appropriate layer thicknesses based on that profile alone. Now that we have these segmentations, we can look at things like cortical thickness, which is an MRI accessible measure, and we can look at it against histologically validated MRI cortical thickness measures from the big brain. So this is what we see on the left and right hemispheres of the big brain. Yellow is thicker, blue is thinner, and you can see that, for instance, the motor strip is very thick, the thickest part of the cortex, whereas the visual cortex is much thinner and is one of the thinnest areas. Through the work of Lindsay Lewis, who has been doing a lot of work registering these surfaces, the big brain surfaces, to template surfaces, we can compare these values with other existing atlases. So on the top right, we have the von Economo cortical thickness atlas. And on the bottom right, we have the MRI in vivo atlas from the Human Connectome project. And what we can see is that the regional pattern of cortical thickness is very consistent between the three data sets. If you're interested in where the discrepancies are, they're covered in the paper, the PLOS biology paper that came out this year. We can go a step further than MRI now because we have this laminar segmentation. So we have here the von Economo measurements for these layers, and underneath the big brain measurements for the same layers, 1, 2, 3, 4, 5, 6, so top and bottom rows. So one thing is that the pattern between the von Economo and big brain is generally very consistent, but there's clearly a lot more information in the big brain because it's not this 40 ROI parcellation. We have per vertex level information about layer thicknesses. And this can be seen quite clearly again with this V1, V2 boundary in layer 4 thickness, where you could basically threshold layer 4 and you would get out a segmentation for V1 because it was really prominent layer 4. So just to summarize, we now have this 3D atlas, pixel-wise segmentation of the big brain and its cortical layers. And this is freely available to download. And one example of how it's been used is by a group in Maastricht who are prototyping their layer fMRI tools. And they've been using the big brain segmentation to test whether their layering algorithms work on a 100 micron volume. We've also got these intensity profiles that capture some of the laminar structure within the cortex. And I think Casey's going to be presenting some of this work, but one example is you can use this to describe microstructural similarity across the cortex, or the gradients of these microstructures, similarity metrics. And then finally, we have these surface-based atlases, which have been registered to template surfaces of cortical and laminar thickness. And one example of how this is being used is by Peter McCoggan at UCL, who is using it to compare with MRI R2 star values, so in vivo MRI data, directly against the big brain staining intensity to see how the microstructure and the MRI structure might relate to one another. So what we're able to do with the big brain is link the microscale histology through the mesoscale laminar resolution and all the way through to in vivo neuroimaging metrics. So that leaves me to thank all the many collaborators that worked on this project, those at the MNI, in ULISH, and also at Mila, the Learning Institute in Montreal, and also my funding from the Wellcome Trust and Healthy Brains for Healthy Lives. Thank you. Any questions?
Thanks a lot, Konrad. We are a bit behind, but I see a question from Famid. So I give you the microphone.
Oh, good. Thank you. Thank you, Konrad, for a great presentation. So I was struck by your statement which leads confidence to the kinds of approaches that we've been using. That identification of layer 4 universally throughout the cortex shows that it appears roughly around 60% of cortical depth. Mm-hmm. However, I feel that that conclusion is really based on the resolution of the data you're working with. So my question is, the laminar profiles that you've been able to identify have really been defined even from the classical works of Zillis, really, is always in 2D. Mm-hmm. But we know that these profiles exist intact in 3D. Mm-hmm. So is there a way to depict laminar profiles, especially with all the cell sign gyrification in the human brain in 3D?
That's, I think there's two things. So one comment was about the resolution, but another one was about whether it's 3D. So with the 3D, the profiles are extracted between vertices, which are in 3D. So the profiles are completely oblique to the original plane of section. They're not done within 2D, which is why we're able to generate these segmentations. So one profile might start in one particular section of the big brain and then end up completely 500 sections further along. So with respect to how this profile-based method with the surface reconstructions basically serves to unfold the cortex and enable you to segment the layers in any oblique direction within the big brain. And with the 60% was for the original layer 4 surface we had. That was somewhere within that dark band of layer 4, but it wasn't clear that it was always in exactly the same place, which is why this full segmentation of 6 layers is now much more comprehensive. So here we have the upper and lower extent of layer 4 now for every vertex across the surface. And as you can see from the layer 4 thickness maps, this does in fact change from region to region.